Hello, in this video we will discuss about the endoplasmic reticulum and Golgi body's function. And we will discuss about their relationship. So the endoplasmic reticulum and Golgi bodies is the organelle, which that is the double membrane organelle, and that is the major organelle in the cell, like a mitochondria. So the rough endoplasmic reticulum and smooth endoplasmic reticulum is we can divide, and rough endoplasmic reticulum contain ribosome where the translation occur, and endoplasmic reticulum is used for protein synthesis and maturation, a maturation as well as the protein maturation and the calcium storage organelle and surrounded around the nuclear membrane. So the smooth endoplasmic reticulum is used for the maturation, and the rough endoplasmic reticulum contain rough. That is why it's contain ribosome. So this is the cell. Basically, let's begin to understand the general structure of the cell. So this is the cell contain mitochondria as well as Golgi bodies, which that is stack-like structure, and this is the nucleus. Nucleus containing inside is a chromosome as a chromatin material for the genetic material. So here is the peroxisome and mesosome, and here is the plasma membrane, and this is the cytoplasm, and there are many things. Here is the cellular matrix, which that is made up of collagen, and there are also the actin and troponin as well as the myosin. There are a lot of things. So here is the chromosome, and endoplasmic reticulum is surrounded by the nucleus. Here is the mitochondria and the cytoplasm, and this is the plasma membrane. So this is the structure. Let's begin to understand the function of rough endoplasmic reticulum and smooth endoplasmic reticulum. So here is the bigger picture for understanding. This is the receptor, G coupled protein receptor, and let's begin to understand the function of the endoplasmic reticulum. So this is that there are different enzymes are also present. Here you can see this is the endoplasmic reticulum, and here is the G coupled protein receptor. When G coupled protein receptor receive the ligand, for example the growth factor, after this the growth factor, the intracellular trimeric protein contain alpha subunit. Trimeric alpha subunit contain inactivation due to the GDP, while the activate due to the signaling by the second messenger enhancement. So the GTP converted into the GDP, and this activation into the alpha GTP or ATP you can see, but GTP. So here is the ATP used to adenylate cyclase enzyme to produce the cyclic AMP, that is the second messenger. After this, the one protein, the phosphoinositide 3 kinase, which that will be activate, and after this activation, it will go to the PIP2, phosphoinositide diphosphate, and in this way, it will be phospholipase C enzyme, which that will be cleaves, and after this, the phosphorylation will be occur to produce the DAG, as well as DAG, as well as the phosphoinositide triphosphate, IP3. This IP3 will be bind with the protein, bind with the IP3 sensitive calcium gated channel, so with the endoplasmic reticulum. After the binding with the endoplasmic reticulum, here is another protein is the activated the protein kinase C will recruit to the DAG after the DAG formation for the further process. We will not understand that things. So here is the IP3 will bind with the channel, which that is the IP3 sensitive channel calcium. So the calcium will efflux of the calcium. So after this efflux of the calcium, it means that calcium will be target the proteins. For example, the calcium will use for the muscle contraction, in the form of and sarcoplasmic reticulum, we can say in the muscle fiber cell, and the protein kinase and phosphatase enzyme is used for it, for target the protein. While here is the DNA, this DNA contains different genes, and this polymerase enzyme used after the transcription with the transcription of factor binding, the specific gene will be activated and transcribe the messenger RNA, and this messenger RNA will bind with the endoplasmic reticulum where the ribosome is present. So the smaller and larger subunit bind together to produce protein. And this protein is not mature, it is a polypeptide, will go into the endoplasmic reticulum inside the lumen. So it will be mature after the further processes. After this immature to maturation to exocytosed or efflux, or you can see the bud off. So the protein will be forming the vesicle. So remember that thing. So let's begin to understand the protein processing and secretion. So the protein processing and secretion we will discuss if. So here is the cell. I am drawing the bigger picture of the effector cell. This effector cell mean it will be affected by something and to release hormone, steroid or protein via exocytosis to for the trigger of another cell is known as the effector cell. So the ribosome assembly and protein from polypeptides in the rough endoplasmic reticulum. So here is I'm drawing the DNA in the nucleus, and after the transcription to the messenger RNA production, this messenger RNA will bind with the ribosome and to form a polysome formation. Polysome means there are many ribosomes will bind with a single RNA, messenger RNA, to produce the copies of the protein with a multiple vesicle formation, because it is like a cloning formation. So the cloning of the protein due to the multiple ribosome attachment is known as the polysome. After this, here is the processing will begin again, and the pinched off this vesicle into the cytoplasm, and after into the cytoplasm, and it will go for the further maturation because it is the semi-matured protein. So let's here is I am writing the protein move through the rough endoplasmic reticulum where they are further modified. So remember one more thing: the rough endoplasmic reticulum is and Golgi body is also used for the lipid synthesis with the protein binding, so the glycolipid formation and glycoprotein. So here is the transport. The vesicles contain the protein are pinched off from the rough endoplasmic reticulum, and in this way the transport the vesicle contain basically the proteins move through the rough endoplasmic reticulum, and the transport vesicle containing the protein are pinched off from the rough endoplasmic reticulum, and Golgi bodies is received. After this Golgi body is received, the further processes will be occur. For the binding of the glucose is known as the glycoprotein. For binding with the lipid is known as the lipoprotein. So lipoproteins will occur in the hepatocyte. So here you can see this is the trans phase and cis phase. Cis phase is receive and trans phase will be pinched off this vesicle. After this pinched off this vesicle, it will remain into the cytoplasm till the primary signal will not arrive. So here is the vesicle travel to the cell membrane and release endocrinally or exocrinally. So it can be synthesized. There are many types. For example, the receptors as well as organelle as well as cell itself repair and use for the synthesis for the set of the organelles, and there are a lot of things for the cell division and cell cycle. So here the vesicle will be formed after the first signal which that is bind. So I am not describing here, but first signal will bind to exocytose due to the calcium increase from the endoplasmic reticulum. Same mechanism, you can understand that well, because the calcium will trigger the most protein for the exocytosis, just like a nerve cell. So the nerve cell is important, as well as the muscle cell will trigger the sarcoplasmic reticulum to release calcium. So that is why, and endoplasmic reticulum is used for the calcium efflux, and there are many things. But this is the neurotransmitter or endocrine system. For example, neurotransmitters. So here is the effector cell will bind this protein endocrinally, and that is the secondary response by autocrinally and paracrinous. Thanks for watching. Please make sure to subscribe. Bye.